Hello everyone, it's me again. Today I'm going to be telling you about how to build a nearest neighbor classifier. We're going to build one up step by step, working with a particular example. And by the way, if my voice sounds a little different today, I'm recovering from a cold, so sorry about that. We're going to be working with an example taken from a high school science fair. This was a project done by a high school student named Brittany Wenger. It was a really cool science fair project. What she looked at in her high school science fair project was diagnosing breast cancer. Okay, so the way this works is if a patient is suspected of potentially having breast cancer, there's a particular biopsy, a procedure where the doctors take a tissue sample, they put it under a microscope, and then they use that to try to make a diagnosis and predict whether the patient has cancer or not. Okay, so Brittany developed a classifier to help make that diagnosis. And it was a really impressive result because she showed how to get 99% accuracy with her classifier. Very cool. As a high school project. Not bad, huh? This thing turned out actually won the National Science Fair project. There you can see her pictured with the president getting congratulated for winning the competition. And to give you a little idea how this works, when you put the tissue sample under the microscope, as a lab tech, the lab tech looking at the microscope sees an image that looks something like this. That's what it would look like if you had benign tissue that was not cancerous. And here's an example of a tissue sample that is malignant, that is cancerous, that shows cancer. And maybe looking at that, eyeballing that, you can get a feeling that those two look a little different. Like there's those clumps, those round circles you see, those are cells, and they're a little bigger in the malignant tissue sample. And you can imagine that an experienced lab tech might be able to recognize by eye some of the characteristics of malignant versus benign samples and use human judgment to try to judge whether a particular sample is malignant or not. Well, that's not the approach Brittany took. Brittany looked at, could we build an automated classifier using machine learning to make the diagnosis so that we don't need as much human judgment, and maybe so we can get better accuracy. So in her data set, the way things are going to work is that a lab tech looks at the image and grades the image based on several characteristics. So there'll be several attributes of this image, like how uniform are the sizes of those cells, those circles? How much are they clumped together, and so on. There's several of these attributes that doctors have guessed as may be helpful in distinguishing between malignant versus benign. And so the lab tech is going to use their human judgment to grade each one on a number from 1 to 10. And then we're going to build a classifier that takes those numbers and uses some prediction rule to predict for each instance whether it's malignant or benign. Okay, so let's take a quick look at the data set. This is the example data set that we're going to use as our basis to build a classifier. We're going to see if we can use the ideas we've taught you to build a classifier and see how well we do, see if we can replicate Brittany Wenger's results. And as you'll see, we don't get a classifier that's quite as good as Brittany. We don't get 99% accuracy. So Brittany still does better than we were able to do with the techniques in this class. She had some more sophisticated methods, but we can get pretty close. So it's pretty neat. So let's look at the data set. All right. What we've got here in our notebook is that we're going to read in the data. You can take a look, eyeball at the data set. It's here as a table, and what you can see is each row represents a patient for which we have historical data. We know whether or not it turned out to be benign or cancerous. The last column here, the class, indicates benign versus malignant. So 0 represents benign, 1 represents malignant, 1 means cancer, 0 means no cancer. And then the other columns are these attributes that are graded by a lab tech. So for instance, here we have uniformity of cell size. Each one of those is a number from 1 to 10. It's somehow judged by a human technician. And they look at the sample and they write down these numbers, these attributes. And given these attributes, we're going to try to predict cancer versus not. So here's a data set. We have data from close to 700 different patients historically. Let's see what we can do with it. Before we start to try to build a classifier or do anything fancy with it, the first step always in data science should be look at your data. So that means we're going to do a little exploratory data analysis. We're going to try to get a feeling for the data. And what I did before I prepared this video was I tried graphing this data in all sorts of different ways. I tried making a scatter plot. I was curious to see how the data for patients with cancer compared to the data for patients without cancer. Okay, so I made a scatter plot. I arbitrarily picked some pair of attributes, and I made a scatter plot with 1 dot per patient. The location of that dot represents the value of these attributes from their tissue sample, and the color of the dot indicates their class, whether that patient turned out to have cancer or not. So the yellow ones are the patients with cancer, and the blue ones are the ones with a benign tissue sample. And what you can start to see from this scatter plot here is that it looks like if you're the patients who fall in the lower left corner of this plot are mostly benign, and the ones who fall in the upper right corner of this plot mostly malignant. So you might start to be thinking, well, low values of bland chromatin, numbers like 1 or 2 or 3, maybe might be indicative of benign, and large numbers like 8 or 9 or 10 might be indicative of malignant, and similar for single epithelial cell size, whatever that is, I don't know, something. Okay. And I started to do this for different pairs of attributes, and yeah, it's interesting. It suggests that these attributes do have some value in predicting benign versus malignant. It's not clear exactly what rule we should use, and as I looked at the data, and as we'll discover as we go on, there's really no 1 or 2 attribute that's enough to do a really good job of predicting. You really need to use all of the attributes to make the most accurate prediction, but just for visualization purposes, we can look at 2 of them at a time. Okay, so that starts to get you a feeling for what's in the data set. Now, if you've been paying close attention, you might notice there's something wrong with this picture. Maybe it's a little unsettling. I mentioned we have data from close to 700 patients. Are there 700 dots in this figure? Doesn't look like it, right? That doesn't look like 700 dots. So what's going on? Where'd all my data go? Why don't I have 700 dots in this figure? Well, it turns out I realized, oh, that's an issue with this visualization. Turns out there are many patients who sometimes they'll have 2 patients that have the same values for the attributes. And if we take those 2 patients and we plot them, the dot will be in exactly the same spot. So 1 dot lands right on top of the other, and you really only get to see one of those 2 dots. So I realized that I wasn't seeing all the dots. Some of these dots are landing on top of other dots that are in the same spot, and I can't see them. So I can't really see how many dots there are. And as I did a little exploration, I realized some of these things, it looks like 1 dot. There might actually be 10 or 20 patients with that value of the attribute sitting right there. It looks like 1, but it's actually 10 or 20. And so I was scratching my head thinking, this is not a very good visualization. How can I make a better visualization that takes that into account and shows me some sense of all the data? And there's multiple different ways you could do that. One way is I could have adjusted this visualization to make a larger dot. If there's multiple patients at this one location, all with the same value, then I draw a larger dot, and the size of the dot is proportional to how many patients have that value. That's one reasonable approach that some people use. I ended up taking a slightly different approach. What I did was I decided I'm going to add a teeny bit of random noise. When I draw a dot, I'm actually just going to shift in a random direction away from the starting point. and then draw it. And that way, if I have two patients with the same values, they'll maybe shift in a different direction and they won't end up exactly on top of each other. So that's what I did. I added a little bit of noise to the data. I took each patient and I added just a little bit small random value to each of the attributes. And then I did the same scatter plot. And now if we do that after we jitter it by adding this noise, we get something that looks like this. Okay, so this hopefully helps you get a better idea of what's happening with these patients and what's going on in this data set. What we can now see very clearly is that, wow, there are a lot of patients with a value down here, maybe bland chromatin of 2 and single epithelial cell size of 2. That might be dozens and dozens and dozens of patients with a benign tissue sample that previously looked like there was just one dot there, but now we realize there's lots of patients down here in the lower left corner. Okay, so hopefully this visualization is a little less misleading. And it also enables you to see that there are some places where, like with bland chromatin of 4 and single epithelial cell size of 2, there were actually about 4 patients, all with those same values. 2 of them had cancer, and 2 of them were benign. So that shows that those patients were indistinguishable at the identical values for these attributes, but had a different class. So yeah, there's we're not going to be able to make a perfect prediction of whether this tissue sample is malignant or benign, at least not by looking at just these 2 attributes. All right, so that's the data set, and what we'll do in our next videos is we'll start working out how to build a classification rule to make diagnosis using this data.